Well, welcome back to evolution. So far, we have reviewed the history of evolutionary thought in Chapter 2, what led up to Darwin's and Wallace's theories of natural selection and common ancestry. Then in Chapter 3, we focused on geology and paleontology because that sets the stage for the history of life, what's occurred here on Earth, and the importance of fossils for showing us the patterns of evolution over the past several billion years. Then in Chapter 4, we looked at how we investigate phylogeny, the history of life, the tree of life, and some of the tools that we use for uncovering that history and answering questions using that history. Now we are ready to confront Chapter 5, which is titled Raw Material, Heritable Variation Among Individuals. So this is really our survey summary of genetics chapter to get us ready for diving into evolution. Because remember, evolution is the change in the genetic makeup of a population over generations, over time. So it's pretty critical that we have down the fundamentals of genetics, where variation comes from, where that variation resides, how that variation is expressed from DNA to proteins, other elements, etc. So an opening story here in the textbook talks about the investigation of human height. And indeed, back in Darwin's day, one could only measure people of different heights and draw various conclusions and interpretations from those phenotypes. But now, of course, we can investigate DNA and the sources of information for controlling such things as human height. So, for example, a survey of almost 184,000 people uncovered 180 different sites in the genome that are associated with height. So we're able to pinpoint where those genes may be located and continue investigating from there. So let's dive into this. Covers quite a bit, and let's get us ready to be able to dive into natural selection and other mechanisms of evolution after we review some genetics. So, of course, the worker molecules of all cells on Earth are proteins, and if one were to take away all the water of most cells, cells are mostly made of water, the dry weight, what's left over after water is taken away, the 50% of that dry weight would be made up of proteins. So half of us organically is protein. And there are 8 functional categories of proteins, such as enzymes, receptor proteins, messengers, structural proteins, defensive proteins, etc. And it's estimated that there's probably about 100,000 different proteins that at least the cells of humans manufacture to build us and maintain us. So we are protein organisms. All organisms are, mostly or 50% dry weight protein. And as you've learned before, it's the sequence of those monomers called amino acids that determine a protein shape. And another one of those themes in biology that always comes back is shape determines function, or structure determines function. And as you've learned, molecular shape is key to how molecules work. So make sure you give a look at the figure here, which is reminding you about how we summarize and describe protein structure. And we do this to empower us to understand what gives a protein its shape so we can understand how it works. So that primary structure is the original sequence of amino acids that make up a polypeptide. The secondary structure includes stretches of those polypeptides that are either folded into what are called pleated sheets or coiled into alpha helices. Then the entire three-dimensional shape of a polypeptide is its tertiary structure. And finally, if a protein is made of two or more polypeptides that are sort of stuck together, then that protein has a quaternary structure. And the whole point of having these levels of structure to learn and remember and refer to is once again because protein shape determines function. And, as I'm sure you remember, the genetic code or the information for making those all-important proteins is found in our DNA. Every cell on Earth contains DNA as its genetic instructions. It's a linear molecule. It's comprised of monomers called nucleotides. And it's the sequence of the nitrogenous bases going down that chain of nucleotides that carries the information for making molecules. The DNA has got two strands twisted around each other into that famous double helix, and the way those two strands match up follows the base pairing rules. Adenines in one strand match thymines in the other strand. Guanines in one strand match the cytosines in the other strand, the base pairing rules. When DNA copies itself to get ready for cells to divide, DNA replication follows from its structure, meaning that those two strands separate, pull apart. Each of the strands serves as a template for making a new strand, a daughter strand. That's called the semi-conservative model for DNA replication. Every new DNA molecule has one old strand, the parent, that was used for copying, and then a new strand that is matched up to it, the daughter strand. So by the time DNA is finished replicating, we now have two DNA molecules. They are exactly the same as each other. They are replicates of each other because of the way DNA copies. Now, changes in DNA, the nucleotide sequence, of course, are mutations. That, by definition, is what a mutation is. And most mutations occur during that copying process when DNA is replicating. So, mutations are the ultimate source of heritable genetic variation. Populations of organisms have to have genetic variation in genes for those populations to evolve. And that genetic variation comes from mutations, changes in the DNA. So the last note here about DNA is just the fact that because all organisms on the planet universally use DNA as their genetic material, but on top of that, all organisms on this planet share a certain set of common genes. I think there's about 20 of them at least, that every life form on this planet has. That's part of our evidence for common ancestry, that we all have a common origin of life. So we'll come back to that. Now this DNA that contains our genetic instructions or information is all coiled up and wound up and found in these compact structures we call chromosomes. And eukaryotic DNA especially is more complexly organized than prokaryotic DNA. And it's very uniquely organized and stored and used and replicated. So for one thing, that DNA is tightly coiled, tightly wound around proteins called histones, histone proteins. And the way the DNA is wound around those proteins is kind of like string being wound around beads. Check out the figure here and visualize that. Look at that. Feel that. So this strategy allows really tight packing of the genetic material into a nucleus so it all fits in there. You might remember that for humans, our DNA is taken out of one nucleus and all the DNA is linearly lined up from all our chromosomes, it would amount to almost 6 feet. So to pack all that DNA into a microscopic nucleus is quite a feat. So this coiling also helps to, besides helping it to fit inside a eukaryotic nucleus, it also helps to hide certain genes inside the coils, making them inaccessible for transcription. So it's one way to help with gene regulation. Or those genes can be made accessible for transcription. But the point is that the coiling helps to control that. So here we're focused on the fact that, as I mentioned on the last slide, DNA is wound up and tightly bundled and packaged into these little discrete packets that we call chromosomes. Now, different species have different numbers of chromosomes. For example, humans have 46, chimpanzees, bonobos have 48, and so on and so forth. But fruit flies have 8. If I remember correctly, horses have like 64. So there's quite a variation in how the amount of DNA is divided up into separate chromosomes. This plant that's shown here, called the adder's tongue, actually has hundreds of copies of each. So pretty remarkable. And of course that becomes an important question in biology when we make observations like this, is why does an organism or a species like this have so many copies? That's what drives us, is answering these questions. Of course, you remember diploid organisms have chromosomes in pairs. Diploid or diploid, meaning 2 chromosomes. And those pairs of chromosomes that have the same genes in them are called homologs, homologous chromosomes. So you could take a look at the karyotype to the left, and it's a human karyotype, so showing you the chromosomes spilling out of a nucleus. And if they're all paired and lined up in what's called a karyotype, then we can see how the chromosomes all match up and occur in pairs. And by the way, of course, above that figure of a human karyotype is showing variations in the numbers of chromosomes or numbers of copies of chromosomes that organisms have. They can be haploid, diploid, triploid, tetraploid, etc. So back to us. In mammals, we've got paired chromosomes in both males and females. And one pair of those chromosomes are the sex chromosomes, 2 X's for females, XY for males. But an important point here, we're not going to get into the nitty-gritty of this right now, but there's a huge variation in sex determination in the animal world. So while most people are familiar with the fact that humans have XY determination, mammals in general, different species of reptiles have other ways of determining sex, and so do amphibians and fish. So there's quite a bit of variation out there. The last point here is, why do chromosomes occur in pairs? Well, in part, we understand that diploidy, having chromosomes in pairs, helps protect against harmful mutations. How is that? Well, if you inherit one copy of chromosome that's got a harmful mutation, and the other copy of the chromosome has a normal, version of a gene, and it's pumping out normal proteins, then in many cases that organism will be protected because it's producing normal proteins, maybe along with malfunctional proteins, a mutant, but it offers some protection to have that normal chromosome with one that's mutant. So there's protection in being diploid. So we've mentioned proteins, most of what makes us up, and the genes and DNA code for making those proteins. We have just quickly reviewed DNA, structure, how it copies itself really quickly, and how DNA is coiled up, found in chromosomes, etc. But now to the next level, evolution's molecules on this lengthy topic, RNA. ribonucleic acid that's used in transcription and in translation. So all organisms, all cells, prokaryotic and eukaryotic, engage in transcription using the DNA code to make RNA copies, and translation using an RNA copy of a gene to then make a protein, a polypeptide, to translate that genetic information, those sequences of nucleotides, into protein information, sequences of amino acids. So RNA is used in both of those processes. There's a whole complex series of steps, as I know you guys have gone over previously. So first we have proteins attaching to a promoter region upstream from a gene. So the promoter region is kind of like a beacon. Hey, this is where we need to start the transcription. So, proteins attach here. So RNA polymerase is the primary enzyme that is attaching to DNA. And as that RNA polymerase is moving down the DNA, the two strands are separating, and the RNA polymerase is helping to copy. Important thing for you to remember here. RNA polymerase is helping to copy the DNA information into RNA information, specifically messenger RNA, carrying the genetic message to make a polypeptide. And then after that messenger RNA attaches to a ribosome in the cytoplasm, that messenger RNA serves as a template for translation. It has carried the information out of the nucleus from the DNA, and it's now a template of information to be used for linking amino acids together to make proteins. So quick summaries that you need to remember and be able to recount without going into all the nitty-gritty of how these processes work. Just an overall summary of what happens, what's going on in our cells, so we have a better understanding of how genetics serves as a foundation for evolution. Onward. So here we're entertaining the topic of gene regulation. So us humans, since having the Human Genome Project in the early 2000s and refining the numbers slightly, humans have about 20,000 protein coding genes in our DNA. Now, just a few slides ago, I mentioned that there are about 100,000 proteins that we make as part of our biology. So you might remember that that number of proteins is made possible by alternative splicing of messenger RNA with introns and exons. And that's mentioned here briefly as part of the slide, as part of gene regulation. So we've got about 20,000 protein coding genes, and about 3,800 of them, almost 4,000, are continuously expressed. So the proteins that are manufactured from these 3,800 genes are used as part of our everyday, every moment maintenance of cells. Then there's about 14,200 genes that are very tissue specific, only being expressed, say, in muscle cell or nerve cell or certain types of connective tissues, etc. It's incredible how we work as multicellular creatures. Gene regulation is absolutely essential for any creatures that are multicellular. And then there's a variety of mechanisms, processes that are used to control or regulate this gene expression. And so check out the table. This table is packed with information. So you should be able to summarize this, though. Basically 5 different stages in which gene regulation occurs. So we have pre-transcriptional regulation, regulation that, for example, coiling or packing of DNA can help make genes accessible or inaccessible for like RNA polymerase to be able to attach to. So that's one type of regulation. Then transcriptional regulation. So DNA is methylated and the binding specificities of RNA polymerase and various types of repressors and activators and so on also help to regulate transcription. So there's a number of different kinds of molecules that control transcription, turning it on or off. Then we've got post-transcription, where RNA is modified, as I mentioned a few moments ago. Introns are removed and exons are bonded together to make a final messenger RNA. So depending on which exons, which parts of the pre-messenger RNA are removed, that is going to determine what the final messenger RNA information has. Number 4, we've got translation itself. And so we can have the binding of regulatory proteins and things like microRNAs that actually regulate the process of translation, even determining whether it's initiated or not. And then finally, once a polypeptide is manufactured from translation, we can have post-translational regulation. So we might have certain amino acid sequences that are cleaved or cut out of the polypeptide, or the binding of other little pieces to that polypeptide, etc. And so that's going to help refine and alter the final structure of a protein, and it might help to activate it, turn it on, or silence it and turn it off. So in summary, there's several different mechanisms or ways that genes are regulated in the production of their molecules. We're still on this topic of gene regulation, and specifically at the level of transcription. So 2 major points here, just 2 walk-away points, is that molecules called repressors can attach to a gene control region and help to repress or turn a gene off. Transcription factors attach to enhancers, and those help to activate gene expression. So gene regulation operates at this level of transcription through repressors and enhancers. So we're continuing with just some of the details of gene regulation, and 3 things are summarized on here. Regulation that happens during translation, post-translation, and then regulation for long-term changes to gene expression. So 3 different levels here. And the main thing I want you to know for translation is what microRNAs do. So of course these come from your DNA, just like all RNA. Take a look at the figure on the top, sort of rightish of your slide. It's showing you some DNA being uncoiled there, and microRNA is being transcribed. And so once that microRNA is prepared and ready, it goes onto the cytoplasm. It binds or attaches to messenger RNA and can block translation. So basically microRNAs silence genes by acting as switches. And it eventually leads to the degradation or the breakdown of messenger RNA. So it's not hanging around in your cytoplasm. Now, microRNAs can even leave the cell of origin, having widespread effects. So there's a little story in your textbook about this with petunias and snapdragons, wild type versus mutant types, and the effects that they have on flower structure. Now, departing translation, post-translation, proteins can be chemically modified by adding certain elements to them. They can be cleaved or cut into smaller little subunits or pieces, and they can be chemically activated. So a protein may not be active or functional right after translation, but post-translationally, we can have some chemical modification to basically turn the protein on. Finally, 2 primary mechanisms for long-term changes in gene expression. Methylation, adding methyl groups to the DNA, helps to silence, turn genes off by blocking transcription factors from attaching. So those transcription factors cannot access the promoter region, therefore a gene cannot be transcribed. Methylation does that. And then also, as mentioned a little earlier, coiling of DNA helps to block genes. So that's more of a long-term strategy for regulating the expression of DNA. You can see we're still on the topic of gene regulation, hugely important topic. First thing that's pointed out here on this slide is how there's a huge range in genome size. So this is table 5.2 in our chapter, variation in genome size and complexity. So there's a number of different organisms listed here. You can see starting on the top, a couple bacteria, then yeast, nematode, thale cress, which is a plant, fly, sea anemone. So good thing you've taken Bio 182. You have some familiarity with this diversity of life. And you can see in the next column, number of chromosomes ranging from bacteria having 1 to the numbers on this chart, dogs having 39. But 39 isn't the most chromosomes organisms have. It can go much higher than that. And then we look at megabases in the genome, and there's a huge amount of variation. You can see the amount of DNA that varies between organism and organism. And yet when you finally look at the last column in that table, approximate number of protein coding genes, you can see that there's not a strong correlation between the size of the genome and the number of genes across species. So something else is going on there besides just the number of genes when it comes to the size of a genome. Another big important question in biology. So in us humans, only a little over 1%, 1.2% of our human genome is made up of protein coding segments. So what's all the rest for? So besides control regions, there's thousands of RNAs that are also made. So that's quite a big chunk, as we've learned, of our DNA is coding for different RNAs. Mutations in DNA can produce what are called pseudogenes, basically ghosts of genes, fossils of genes in our own genome. They're not used, they're not active. They can't be taken out, they're stuck there, they get copied every time our DNA is copied, but they're not functional genes. There's over 17,000 of them in us humans. Remember, we've got 20,000 protein coding genes. Over half of our genome is made up of what are called mobile genetic elements that can copy themselves and insert themselves in other parts of the DNA. So there's far more going on in our DNA than just protein coding genes. So after all that business with genes and gene regulation and other components of our genome and what they do, we now are to the point of talking about where variation comes from, genetic variation comes from. Of course, as already mentioned, mutation, changes in the nucleotide sequence, changes in the DNA. Now, these changes can happen randomly during DNA replication. You should remember that when those nucleotides are matching up to make daughter strands using parent strands, by random chance, there might be a mismatch. And if those mismatches are not detected and fixed, which eukaryotes are pretty good at doing, then those mutations are going to stick. Also, radioactive particles can cause damage to DNA, cause mutations, radiation even from the sun, ultraviolet radiation, or chemicals called mutagens may interfere with DNA replication and also cause mutation. So, but the most important source is random errors during DNA replication. So the figure to the right is summarizing for us ways in which mutations may happen. So point mutations, just a single nucleotide changing in the DNA, most common ones being substitutions. One nucleotide being substituted with another. But we can also get insertions and deletions, having new nucleotides that weren't there before, fitting into places that weren't occupied by nucleotides before. That would be an insertion. Or taking out nucleotides. They're not there anymore, and the daughter DNA, those would be deletions. Gene duplications. These are less common. These are almost sort of listed in probability of happening. So genes might be duplicated, say, during meiosis, actually, when crossing over occurs. If there's unequal crossing over, then a chromosome could have a gene duplicated. We can have inversions. So we can have genes basically being flipped around. So that's possible, or parts of a gene flipped around. Chromosome fusion. Could have two chromosomes that fuse together and make one chromosome. This happened in the humans after we branched off from a common ancestor with chimpanzees and bonobos. Remember, I said a little earlier, they have 48 chromosomes. They have 24 pairs. And we have 46 chromosomes. We have 23 pairs. We know exactly which chromosomes fused together in our ancient human lineage that reduced us from 48 to 46 chromosomes. It's a rare event, but it does happen. And even more rare is genome duplication. So an entire genome was duplicated, producing entirely new sets of chromosomes. Now, mutations can be harmful. They usually are, or helpful. They might confer some benefit in terms of natural selection, or neutral. They don't do anything either way. But no matter what, mutations always result in new genetic variation among individuals in a population. And remember also that if a mutation in one environment or context is harmful, in another environment or context, it may be beneficial. So mutations, whether they're harmful or helpful, may be context dependent. Finally, mutations can change how much protein is made, or when it's made, or the location that proteins are made. So it's not just all about a mutation producing a new kind of protein, a new variation of protein, but as noted here, how much of it, or when, or where it's produced. So here's some nifty little graphs to check out. We're still on the topic of mutations, creating new variation. And now these graphs are showing you, the upper graph is showing you, out of these different categories of mutations, which ones are the most common and which are the least common. And as you can see, point mutations have the highest rate of production of all the categories of mutations in humans. But this applies not just to humans, but this is reflecting humans. And then point mutations are followed by small insertions and deletions, and then insertions of mobile elements that are jumping around, then large duplications and deletions, and finally gain or loss of chromosomes. So that's the sequence or the order in terms of the most common types of mutations to the less common. Now, if we look in part B, or figure B, it's showing you something different. Total base pairs of mutations contributed by each category. So when you look at it from this perspective, point mutations, although they are the most common type of mutation, they don't have the biggest impact on DNA structure. They're kind of in the middle there. You can see gain or loss of chromosomes ends up being the biggest impact on the total number of base pairs, mutations. Okay, so although gains and losses of chromosomes is pretty rare, when it does happen, I've already mentioned, it has a huge impact, huge impact. Now, interestingly, each newborn human has about 61 new point mutations. So we're born with, new unique mutations, all of us, all the time. Now, every 42 births, there's a large duplication, greater than about 100,000 bases. So it's pretty dramatic when you learn about it and realize it, that every time we reproduce, there's quite a bit of change to our DNA. It's not staying the same. So now we're turning our attention to heredity. We've covered structure, DNA, talked about RNA, talked about how gene expression is regulated. We've just talked about where new genetic variation comes from, primarily mutations, different kinds of mutations that happen and their impact. So now heredity, the passing of genetic material from parent to offspring. That's heredity. So compared to prokaryotes, sexually reproducing multicellular eukaryotes, that would be us, that would be plants, to some degree fungi, some types of protists, we exhibit extremely complex heredity compared to prokaryotes. So, of course, we have homologous chromosomes, or pairs of chromosomes from separate parents. And each parent is giving us a subset of their own alleles. And so we have many, many, many different alleles to choose from different parents. So that's a major component of eukaryotic heredity, is that we're getting chromosomes from different parents. And these different parents have many different alleles. Prokaryotes do not do that. Now, during meiosis, which is necessary for sexual reproduction to reduce the number of chromosomes by 1/2, so that each sperm cell or each egg cell has 1 instead of 2 chromosomes of each type. During meiosis, genetic recombination occurs by a crossing over. So remember, that happens during prophase 1 of meiosis 1, where homologs come together and synapse and go through crossing over and swap pieces of their chromatids. And that helps to mix up combinations of alleles. That does not make new alleles. It makes new combinations of alleles along chromosomes. Then, independent assortment. That's where homologs are randomly lining up on either side of a metaphase plate. And each pair of homologs is doing this independently of all the other pairs of homologs. It's independent assortment. And because of that random placement of homologs like that, that also produces genetic variation in gametes. So not just crossing over, which is prophase 1, but then metaphase 1 is now involved with that independent assortment. And that's important for producing variation during heredity. So each parent, each male, each female is able to produce millions of genetically different combinations from meiosis. And out of those millions of genetically different combinations, certain gametes randomly combine, more or less by chance, to produce a zygote, a new offspring. So these figures that I've provided here are summarizing for you some of what happens in males and females during meiosis, stuff that you would have gone over in Bio 181. And again, we're just going over the generalities here. So focus on these highlights that I'm summarizing for you to just make sure we've got this nice foundation or base about genetics and variation and heredity to lead us forward into evolution. So we just gave some attention to what happens in sexually reproducing eukaryotes. Be fairly complex. A lot of variation is produced through those processes in meiosis and then the random combination of gametes. But what's going on in bacteria and archaea? What's going on in the prokaryotes? Again, you guys probably have some background in this, but remember, they have a single circular chromosome, not linear chromosomes like eukaryotes have, but circular chromosomes that sort of floats around within a cell. It's not located in a membrane packet, a nucleus, but it's just kind of free inside the cytoplasm of the cell. Now, in prokaryotes, they don't have any histones. So there aren't any proteins that are keeping the DNA sort of twisted into loops. They don't have that way to sort of package up the DNA and regulate the DNA with histones. That's unique to eukaryotes. There are, however, genetic regulatory regions upstream of genes. Those respond to the environment. And so that is one way that prokaryotes are regulating the expression of their genome, is through those. So those are some differences, but here's some more. Prokaryotes lack enhancers that regulate gene expression. Eukaryotes have them, prokaryotes don't. Prokaryotes lack alternative splicing. So they don't have introns that are excised or cut out and exons that are bonded together. Don't have that. So they don't have alternative splicing that limits their how much their genome can be used for making different products. And finally, prokaryotes lack the variation-producing mechanisms of sexual reproduction. They don't do meiosis. So they don't do crossing over, they don't have independent assortment, they don't have the random fusion of gametes. So you look at this whole list here, there are prokaryotes are much more limited in the way that they can express their DNA than eukaryotes are. There are profound similarities, of course, but these are some very important differences. Now, when we compare the DNA of the actual DNA of prokaryotes and eukaryotes, just the raw DNA, it's profoundly similar. And prokaryotes have many of the same kinds of mutations as eukaryotes do. So point mutations, insertions, deletions, errors that happen during their own DNA replication, they have plenty of those as well. Because prokaryotes are able to reproduce so quickly and have a fast turnaround time in the division of their cells, beneficial mutations can spread through a bacterial or a prokaryotic population very rapidly. Because if there's a mutation that's beneficial, which evolutionary mechanism is going to cause that beneficial mutation to spread and become more common? Good old natural selection. So prokaryotes are subjected to that as well. Of course, so are viruses, on a side note, thinking about the coronavirus variants that are popping up, that our vaccines are less targeted against. So that's a selective benefit for these viruses, be able to increase transmission rates, etc. So we're not even talking about viruses here, by the way, but I'm just relating to you that genetic mutation and natural selection are rampant across the board between both the true living world of cells and the quasi-living world of viruses. Now back to what I'm talking about here. In eukaryotes, the transmission of DNA is primarily vertical, which means across generations from one cell or one organism to another. But in prokaryotes, they have vertical transmission as well, of course. As cells divide, they vertically, through time, pass their genetics onward. But prokaryotes also have horizontal transmission, meaning from one cell to another cell, without the cells dividing and reproducing, but just like, hey, here you go, here's some DNA, have it, it's free from me. So via plasmids, which are the little circular pieces of DNA that can be transmitted from one prokaryote to another. So that's a very important difference between prokaryotes and eukaryotes in terms of heredity, is that horizontal transfer of genetic material is much more common in prokaryotes. Now, this kind of transmission, of course, can be helpful. So like with antibiotic resistance, a lot of bacteria out there have picked up genes from other bacteria horizontally that have made them resistant to antibiotics. But there's also horizontal transmission that can be harmful, and it could lead to death of the cell. So it's quite a range of consequences. So here, little transition, we're starting to bridge the gap, if you will, make the connection between the genome, the genetics, and the phenotype, what's expressed. And there's, a very complex link between most phenotypes and genotypes, and it's been quite a challenge and struggle for geneticists to pinpoint and find those links, but we are doing it. So here on this slide, it's showing you a pretty simple link using this plant called the ivy leaf morning glory, and it provides a good example of discrete variation. Discrete variation is sometimes referred to as either or variation. So you have one gene with two alleles, and based on those two alleles, those two variations, an organism can either come out with one phenotype or another phenotype. Either or. That's why it's called either or variation. Discrete means like binary, very, like oppositely different, if you will. And this is usually based on one gene with two alternative alleles. So in this figure, these alleles are symbolized as uppercase T and lowercase t. So you can see in these diagrams how we can get the uppercase T's are coding for lobed leaves and lowercase t for unlobed leaves. And so with simple Mendelian genetics, we end up with two different leaf types. They look pretty dramatically different, pretty dramatic shapes, but it's just one gene coding for a protein that is determining this difference in the phenotype. Discrete variation. One gene, two alleles, simple Mendelian inheritance. And how could we possibly pass through the discussion of heredity without bringing up good old Gregor Mendel, the father of genetics, the father of figuring out the foundations of inheritance. And remember, Mendel, it's one of the few history stories you learn about in Bio 181. But Mendel worked in the mid-1800s working with pea plants because they provided a good model organism to answer the questions he was curious about with genetics and inheritance. He happened to pick the right plant and picked some of the right genes to elucidate or make inheritance clear. So it was only, through studying crosses made up of many different plants could he see the 3-to-1 ratios of traits that eventually led him to infer or conclude that there were dominant alleles and recessive alleles, that genes occur in pairs and an organism can inherit two of the same alleles. They can be homozygous, or they can inherit two different alleles and be heterozygous. And these alleles can be dominant or recessive, and that can control the expression of the alleles. Okay, so a summary of some of the things that Mendel discovered. But remember how we did this? So for that, we've got this figure, shows a lot of information on the right-hand side of the slide. So we start off with two parents that are homozygous for a particular gene. In this case, the gene that codes whether a pea is going to come out smooth or wrinkled. So you start off with a parent who's homozygous for the allele for being smooth, and another parent who is homozygous for the alleles for being wrinkled. Now, these parents produce gametes through meiosis. Parent number one only produces gametes with the allele for being smooth, and parent number two is only producing gametes with alleles for making wrinkled seeds, because those are the only alleles the parents have. So the F1 offspring, all are going to be genetically identical. They're all going to get one big R from one parent and one little r from the other parent. And this is what Mendel saw in his genetic crosses, if you remember, like with flower color and the height of the plant and all kinds of things, including round versus wrinkled. So all the pea plants in the F1 generation showed smooth seeds because they all inherited a dominant allele from that one parent. But then it's what happened in the next generation that really revealed to Mendel how inheritance works. Because even though these F1 offspring all had smooth seeds, when he mated those, when he crossed those, he ended up with the F2 offspring, and he inevitably got in this case, 3 out of the 4 offspring had smooth seeds, and 1 out of the 4 offspring had wrinkled seeds. So it was these results that led him to conclude how inheritance must work. It was seeing these patterns of inheritance over and over again that led him to conclude that, okay, organisms have chromosomes in pairs, they're diploid, and when organisms make gametes, each gamete only gets one of those chromosomes, goes into one gamete, the other chromosome in the other gamete, segregation. And then those gametes randomly combine, and an organism could get a dominant allele and a recessive allele, or 2 recessives, or whatever. So he came up with those 3-to-1 ratios. So this was groundbreaking, right? I mean, Mendel figured out how inheritance works. And that theory of inheritance had profound ripples all through biology, including for evolution. So, by the way, for those wrinkled seeds, what's going on there, those plants that have that allele, the little R recessive allele, they don't make functional copies of a starch branching enzyme. It's got a little, name there, SBEI, and it helps to break down sugar. That's what that enzyme does, helps break down sugar. So with those pea seeds not being able to break down sugar, the seeds absorb water via osmosis from the environment, and they swell up. And then later, when the seeds dry out, they shrink and the surface of the seeds folds and wrinkles. So that's the mechanism. That's what's causing wrinkled seeds. And you've got to have 2 copies of that allele for making a non-functional enzyme. And so as long as you inherit 1 copy of a functional allele, a big R allele, you will make that enzyme and your seeds will come out round. You've got to inherit 2 recessive alleles, 2 dysfunctional copies of that gene, and that's the only way you can get little r, and get wrinkled seeds. So not only do we understand the mechanisms of inheritance, what happens through Mendel's principles, but we also understand like the biochemistry of what's going on that actually causes seeds to be round or wrinkled. Now here's a concept you may or may not have learned about before. I don't know. It's called polyphenisms. Move myself down there. So what is a polyphenism? A polyphenism is a phenotype that doesn't correspond to black and white alternative genotypes. And so the reason for that is there's environmental signals that cause the exact same genotype to be expressed in different ways. So regulatory signals from the environment that are basically controlling gene expression. And this is very often due to many genes that are interacting with each other and the environment at the same time. That's how we get many polyphenisms is through these interactions. So an example used by your book is with these beetles and whether they grow horns or not. So that is dependent, the horn growing ability is dependent based on their nutrition. They will only grow horns if they are able to garner enough nutrition. So you can see a couple graphs that are included here as well. Adult body size. So for smaller adults, they tend to not have horns or very short horns. And then you see very quickly in that graph, the horn length increases quite dramatically with just a slight increase in body size. And so that's all correlated with growth and with nutrition. So again, the overall concept, polyphenisms, phenotypes that don't correspond to alternative genotypes because the environment is having an influence. So here's a little bit more about genes and the environment. Remember a little bit earlier, I was talking about discrete variation, either or variation. Either a phenotype is A or a phenotype is B. And that's usually controlled by one gene with two alleles. So the simplest example. Francis Galton studied continuous variation. This is referred to as quantitative genetics or quantitative traits. And we're going to look at that in a future chapter, the evolution of quantitative traits and how we can study that evolution. Now, Galton studied continuous variation in human height, and he showed a normal distribution in height, which you can see on the graph here. So the shortest people occur in the very lowest frequencies, and the tallest people also in the lowest frequencies. And when you get towards the mean or median or whatever height, that is the most common of the heights, has the greatest frequency. And so this results from the interaction, the sum of variation caused by genetics, usually numerous genes to get this broad variation, and also influences from the environment. So for example, one good clear example that's talked about in your textbook is the Maya people of Guatemala. They're generally known for being short, and it was once thought that that was completely caused by genetics. They had unique genetics, but in fact there are environmental influences that cause them to typically have a shorter stature, nutritional sorts of variation. So Galton is one who studied variation in humans, variation that shows a continuous pattern, and that is caused by the interaction of genetics and the environment. So here's a good way to look at the influence of genetics on human height. Compare fraternal and identical twins, because of course identical twins come from the same zygote, so they have exactly the same genome, and fraternal twins come from two zygotes, two separate sperm cells, two separate egg cells. Are essentially the same as siblings that are born at different times. So here we have a couple plots on the page, a couple graphs, and the top plot is of identical twin heights. So you can see when we compare the height on the x-axis of twin number 1 to the height of twin number 2 on the y-axis, boy, that's a beautiful line there. So monozygotic twins, genetically identical, very clear relationship being shown there between genetics and height. Whereas when we look at dizygotic twins, twins that come from two zygotes, they are not genetically identical, just like siblings born at different times, then you can see there's not a clear connection. So genetics is not explaining their similarities in height. They actually have quite different heights. So when you plot the height of twin 1 to the height of twin 2, you get a little bit more of a scatter plot. You can have a line through there, but it's a much less clear pattern. Now we're going to talk about QTL later on in the book, quantitative trait locus analysis, It's a way of finding where in the genome where to find the genes that are responsible for complex genetic traits. And so we've been able to do that with humans and to use this QTL analysis to find which genes are involved with human height. And so, yeah, we'll check that out a little bit later. Now is not the time. But the point is that we can locate these genes in our genome and find where they are, and then study them more closely to see what their influences are. All right, so continuing, QTL for height. So there's a little bit more here. There's an LOD score of 2, indicates that a genetic marker is 100 times more likely to be associated with a height-related gene than not. So look at the graph and look at figure A on the top. So LOD score is on the vertical axis. Location on the human map is on the x-axis. So that's where, in the human genome, on which chromosome and what part of the chromosome genes might be found that are influencing, in this case, human height. And so for, you can see on the x-axis 1.7, 3.3, 4.8, so that's telling you which chromosome and where on the chromosome. And you can tell by those scores, remember what was just said, statistically speaking, an LOD score of 2 or higher indicates this strong relationship with height. So you can see, looking across the genome there are a couple different places that are hitting an LOD score of 2. Now, if you look down at graph B on the bottom of the page, it provides a closer look at chromosome number 6. And if you look at chromosome number 6, specifically going across there, you can see, bloop, there's a strong LOD score right about in the middle of the graph there. So that's showing you that there is a genetic marker there on chromosome 6 that is linked to human height. All right, so we're wrapping up this chapter that's doing a whirlwind tour of genetics, where genes are found. Again, we start out with DNA and RNA and how genetics is expressed and how it's regulated, where variation comes from. There's a lot of key important topics we've gone over here. But to wrap this discussion up, how do genes respond to the environment? Now, for a gene, from a gene's perspective, the environment refers to anything, whatever, might influence its regulation. So we're not talking about the environment just as something outside of the organism, but the environment of the gene within a cell. That's the environment. And so it could be an external factor. It could be, for example, light might have an impact, but it's more likely to be an internal environmental factor, like a hormone, for example. Now, morphogens are molecular signals that sort of move through a field of cells, such as embryos. And so they are an environmental influence that's having an impact on genes. And that's what these figures here are depicting or showing you, is how morphogens are moved through a very early embryo, or egg to embryo of an insect, and influence the spatial patterning of how genes are expressed. So a very important part of gene expression in animal embryos that determines which cells are going to be turned on and turned off to make various body parts. Here's another concept that we're going to come back to in a later chapter called phenotypic plasticity. So that refers to a range of possible variation given one genome. And this is due to environmental influences. This is called phenotypic plasticity. So, for example, I'm able to get a suntan. I have naturally, fairly light skin, but if I spend a lot of time out in the sun, then I produce more melanin as a protective mechanism and get a suntan. So that's an example of phenotypic plasticity. There's a range of variation that can be expressed based on my environment. So it's usually an adaptive strategy that's used to tailor phenotypes to an organism's environment. So phenotypic plasticity is adaptive, and there's a genetic basis for phenotypic plasticity. So I use the example of, say suntan, melanin production from exposure to the sun, and indeed that is adaptive. To not have that protection from ultraviolet radiation means more DNA would be damaged in my skin cells, etc. So we'll return back to some of these concepts in later chapters at appropriate times in discussing evolution. But this rounds out our chapter on genetics and important principles in genetics and in heredity that we need to have as a strong foundation or base moving forward into our next chapters in specific topics of evolution.